today's topic is molecular basis of inheritance. In that, RNA world and replication of DNA will be discussed in today's program. Regarding the genetic material, DNA is the main genetic material, whereas RNA assists DNA in protein synthesis or expression of Mendelian characters, and except in RNA viruses, DNA is the genetic material. Because of this, the probability of RNA also acting as a genetic material, the question arises, which is the first genetic material? Is it RNA or DNA? Which is the first genetic material? If we look at cellular processes, important cellular processes like protein synthesis. Protein synthesis is centered around rRNA, tRNA and also mRNA. And proteins are essential for metabolism, and different kinds of synthesis processes take place due to catalytic activity of proteinaceous enzymes, but the proteins themselves are formed due to RNA enzyme called ribozyme. The 23S rRNA is essential for synthesis of proteins. So, it catalyzes the bond between two amino acids. So, it catalyzes the peptide bond between two amino acids, thus forming the polypeptide or protein. So, the important cellular processes are centered around the RNA, and because it is the first genetic material, RNA is first genetic material because it is unstable and it can easily undergo mutations. From RNA, from uracil we have thymine, the 5-methyl uracil. So, by chemical evolution, RNA changed into DNA. RNA is the first genetic material. From RNA, DNA is evolved. It is stable. It resists change, faster change, due to presence of repair mechanism. And because the ancient nature of RNA being genetic material is supported, that hypothesis was supported by the ribozyme enzymes and a splicing process in eukaryotic organisms. RNA లేదా DNAలలో ఏది మొదటి జన్యు పదార్థం అనే దాన్ని పరిశీలించినప్పుడు రెండు RNA, DNA రెండు ప్రతికృతి చెందగలవు. మరి రెండింటిలో ఏది మొదటి జన్యు పదార్థం అని నిర్ధారించడానికి ప్రత్యక్ష సాక్ష్యం లేకపోయినా శాస్త్రవేత్తలు కణంలో జరిగే ముఖ్యమైనటువంటి జీవచర్యలు దేని ఆధారంగా జరుగుతున్నాయని గమనించినప్పుడు ప్రోటీన్ సింథసిస్, ప్రోటీన్ల సంశ్లేషణకు rRNA, tRNA, mRNA మూడు కూడా అత్యవసరం. ప్రోటీన్ సింథసిస్ మొత్తం యాంత్రికమంతా కూడా RNA చుట్టూ నిర్మితమై ఉంది. అలాగే ప్రోటీన్ ఎంజైముల వలన రకరకాల జీవచర్యలు సంశ్లేషించబడతాయి. ప్రోటీన్ల సంశ్లేషణకు RNA ఎంజైము 23S rRNA రైబోజైమ్ అత్యవసరం. దీని వలన RNA మొదటి జన్యు పదార్థంగా శాస్త్రవేత్తలు భావిస్తారు. రసాయన పరివర్తన వలన RNA నుండి DNA ఏర్పడింది. యురాసిల్ 5-methyl uracilగా మారింది. థైమిన్ ద్విపాచ నిర్మాణం ఏర్పరచుకున్నది. అదే రకంగా స్ప్లైసింగ్, mRNA స్ప్లైసింగ్ నిజకేంద్రక జీవుల్లో hNRNA స్ప్లైసింగ్కు లోనై mRNAగా మారటం కూడా ఈ విషయాన్ని నిర్ధారిస్తుంది. Now let us consider replication of DNA. DNA is a double stranded. Its double helical structure was proposed by Watson and Crick. Watson and Crick, while proposing double helical structure of DNA, also suggested a mechanism for replication of DNA. The double-stranded DNA unwinds and each strand of dsDNA will be acting as templates. So, this is the double-stranded DNA and it unwinds and each strand will be acting as a template. Each strand is acting as a template, can synthesize the complementary strand by rule of complementarity. So, this will be acting as a template, it will synthesize another strand and this will be acting as a template, will synthesize another strand and identical copies being produced in the new two daughter strands formed. One is old or parental, other is new. So, two older parental strands, two daughter strands are there. This kind of replication is called semi-conservative method of replication. Semi-conservative method of replication means half old strand, half new strand, half old strand and half new strands. Without any experimental proof, Watson and Crick proposed semi-conservative method of replication for DNA. What is the basis for their such proposal? What is the basis for their proposal? A and B, A and T are bound to each other. A of one strand is bound to T of other strands, complementary strand, and the two are held by two H bonds. Similarly, G of one strand is held to C of the other strand and the two are bound by three hydrogen bonds. So, basing on this, because there is fixed complementarity, automatically this acting as a template will regenerate the other, this acting as a template will regenerate the other strand. Watson and Crick proposed semi-conservative method of DNA replication without any experimental proof, but it appears to be logical. The actual proof was provided by two scientists, Michelson and Stahl. Michelson and Stahl using N15 ammonium chloride, they have grown E. coli cells, Escherichia coli cells in N15 ammonium chloride for many generations. As a result, the entire nitrogen content in the E. coli cells is made up of N15 only. Then, from some cells, they have extracted DNA and subjected that to centrifugation in cesium chloride solution. The DNA was subjected to ultracentrifugation in cesium chloride solution, and in the centrifuge tube, the DNA appeared at the bottom. The DNA appeared at the bottom. This is made up of N15 and N15. Later, these cells were transferred to N14 NH4Cl medium. They were allowed to grow. After some time, the DNA is extracted from the cells and it is subjected to centrifugation, and say after 20 minutes they have separated some cells, they were broken, their DNA was taken in cesium chloride and subjected to ultracentrifugation, and in the centrifuge tube, the DNA appeared in the middle because it is not heavier, So, this is N14, N15 DNA, hybrid DNA, because it is heavier one, it appeared at the bottom, because it is intermediate in weight, it appeared in the middle. And they allowed one more replication of the remaining live cells. In N14 medium, and they extracted their DNA and subjected to centrifugation in cesium chloride. And they observed two bands, one intermediate band and one lighter band in the centrifuge tube. They observed one lighter and one intermediate bands of DNA in the centrifuge tube. So, basing on this experiment, they concluded that the DNA replication occurs in semi-conservative method. What happened in the first one? When E. coli cells were grown in heavier nitrogen, that is N15NH4Cl, the nitrogenous compounds, the DNA made up of nitrogenous bases, this heavier nitrogen was incorporated into its DNA, and the heavier DNA was obtained at the bottom. N15. Then the live cells were transferred to N14NH4Cl medium and allowed to replicate once for about 20 minutes. Then the cells are separated and their DNA was extracted and subjected to ultracentrifugation in cesium chloride. And here each strand acting as template synthesized a new strand. As a result, hybrid DNAs are produced. And this is heavier. N15 nitrogen is heavier, whereas N14 is lighter. The hybrid appeared in the middle, and later the remaining live cells were allowed to replicate one more time, and the cells are killed and their DNA was extracted, and in cesium chloride it is subjected to centrifugation. Two bands appeared here, and the lighter one and the intermediate one. The two strands separated. So, these are the two strands separated here. Acting as templates, they synthesized the complementary strand. So, this is the N15 strand, and let me draw the N14 strand here. And this has synthesized a N14 strand. So, this is N14, and this is N15, N14. Now, let us consider this. The two strands separated, acting as templates. So, this is the N15 strand. This acting as template synthesized a new strand. So, this is N15, this is N14, and this acting as a template synthesized a new strand. So, this is N14, N14. These two appeared as a lighter band here. these two appeared as intermediate weight. DNA ప్రతికృతి అర్ధ సంరక్షక విధానంలో జరుగుతుందని వాట్సన్ క్రికులు ప్రతిపాదించిన ఏ రకమైన ప్రయోగాత్మకంగా చూపించిన రుజువు లేదా సాక్ష్యము లేదు. మెసల్స్ అండ్ స్టాల్ శాస్త్రవేత్తలు ఈకోలై సెల్స్ను ఈకోలై కణాలను భార నత్రజని N15 NH4Cl కల పోషక యానకంలో వృద్ధి చేశారు. అనేక తరాలు వృద్ధి చేయటం వలన ప్రతి నత్రజని సంబంధిత పదార్థము N15 కలిగి ఉంటుంది. భార నత్రజని కలిగి ఏర్పడింది. ఈ కణాల నుంచి డిఎన్ఏను సేకరించి నిష్కర్షణ చేసి అల్ట్రాసెంట్రిఫ్యూగేషన్ అవసాదనం చేయగా సీసియం క్లోరైడ్లో అవసాదనం చేయగా అవసాదన నాళికలో అడుగు భాగాన అధిక బరువు వలన అడుగు భాగాన ఈ డిఎన్ఏ చేరింది. తర్వాత ఈ కణాలను సజీవ కణాలను కొన్నింటిని NH4Cl N14 రకంలో వృద్ధి చేసి మరల కణముల నుండి డిఎన్ఏను సేకరించి సీసియం క్లోరైడ్లో అవసాదనకు గురి చేయగా మధ్య భాగంలో ఈ డిఎన్ఏ పోచ ఏర్పడింది. మధ్య భాగంలో ఏర్పడటానికి కారణం ఇది N14 N15తో తయారవటం వలన. ఇది క్రింది భాగంలో ఏర్పడడానికి కారణం అధిక భారము. రెండు పోచలు కూడా N15తో ఏర్పడి ఉండుట. మరలా మిగిలిన సజీవ కణాలను ఇంకొక పర్యాయము ప్రతికృతి చెందినట్లుగా చేసి డిఎన్ఏను సేకరించి అవసాదనకు గురి చేయగా అల్ట్రాసెంట్రిఫ్యూగేషన్ సీసియం క్లోరైడ్లో చేయగా రెండు బ్యాండ్స్గా కనపడింది. ఒకటి N14-15 మధ్య స్థాయి భారము, తేలిక తక్కువ భారం కలిగినటువంటివి ఏర్పడినాయి. తక్కువ భారం కలిగినవి N14తో ఏర్పరిచిన పోచలు. మధ్యస్థంగా ఉన్న భారం కలిగినవి N15-14తో ఏర్పడిన పోచలు. ఇవి అర్ధ సంరక్షక విధానంలో ప్రతికృతి చెందినప్పుడు రెండు పోచలు విడిపోయి కొత్త పోచను పాత పోచ మూసవలే వ్యవహరించి కొత్త పోచను తయారు చేసుకోవటానికి తోడ్పడతాయి. సో పాత పోచలలో N15, కొత్త పోచల్లో N14 ఉంటాయి. దానివలన మధ్యస్థ భారం కలిగినటువంటి బ్యాండ్ ఏర్పడింది. తరువాత మరల ప్రతికృతి చెందినప్పుడు ఈ పోచలు విడిపోయి ప్రతిదీ మూసగా వ్యవహరించి ఈ పోచ కొత్త పోచను N14 పోచను తయారు చేసుకుంది. అదేవిధంగా ఈ పోచ N14 పోచను తయారు చేసుకుంది. ఈ విధంగా ఏర్పడటం వలన రెండు బ్యాండ్స్ ఏర్పడినాయి. సో అండ్ ఇన్ prokaryotic cells this is proved as a semi-conservative method. Taylor and associates working on faba bean 𝗧𝗦𝗔𝗧 they proved that the DNA replication in eukaryotic organisms also in semi-conservative method. టైలర్ మరియు అనుచరులు ఫ్యాబా బీన్గా పిలువబడే 𝗧𝗦𝗔𝗧 లెగూమ్ మొక్కలో డిఎన్ఏ ప్రతికృతి అర్ధ సంరక్షక విధానంలో జరుగుతుందని రుజువు చేశారు. కేంద్రక పూర్వజీవుల్లోనూ నిజ కేంద్రక జీవుల్లోనూ రెండింటి యందు డిఎన్ఏ ప్రతికృతి అర్ధ సంరక్షక విధానంలోనే జరుగుతుందని రుజువు చేయబడింది. Now let us consider one numerical problem. This is the problem. N15 DNA was allowed to replicate in N14 medium for 3 generations, and we have to calculate how many of the following will be there: the number of N15 strands at the end, the number of N14 strands, the number of hybrid DNAs having one strand of N15 and other strand of N14. DNAs, heavier DNA are DNAs with both strands made up of N15, and DNAs with both strands made up of N14 are new type. The DNA replication, so the heavier DNA 15-15 type DNA replicated in a medium of 14-14. So, the dotted line represent 14-14, continuous line represent 15-15. So, at the end of first replication, 2 DNA strands are there, and this is first replication. Now, second replication, these two are acting as templates. So, these 2 strands of each DNA acting as template. Now, let me draw this acting as a template, synthesizes one. This acting as a template will synthesize another. This acting as a template will synthesize another strand. This acting as a template will synthesize another strand. So, at the end of second replication, we have 4 DNA strands, and at the end of third replication, each will be acting as template. As a result, we have, so how many strands are here? 1, 2, 3, 4, 5, 6, 7, 8. So, 8 templates are here. Let me draw here, 8 templates will be there. The first one will be this, second, third, fourth, fifth, sixth, seventh, and eighth. Each synthesizes a new strand. So, we have 8 DNA molecules with us, 8 double-stranded DNA molecules with us. So, at the end of third replication, we have 8 DNA molecules with us. So, total number of DNA molecules formed at the end of third replication is 8. This can be concluded like this. Replications are 3. So, 2 to the power 3, 8 new DNA molecules will be there. 8 new daughter DNAs will be there. At the end of first replication, 2 hybrids are there. At the end of second replication, we have 4 DNA molecules. In that, 2 are hybrids and 2 are new ones. At the end of third replication, 8 DNA molecules are there. Now, let us try to find out the first question. Number of N15 strands is only 2 are having N15 strands, because only original DNA, this is changed into the N15 DNA is changed into N14 medium. So, only 1 will be there. So, 1 DNA, that is its strands are separated. So, for the first one, our answer is 2 DNA strands will be there. And how many strands are there in the total 8 DNAs? 8 into 2, 16 strands are there. Out of these 16, 2 are N15 type. And how many N14 strands are there? Out of 16, 2 are like this. Remaining 14 will be N14 type of strands. Next, number of hybrid DNAs. How many hybrid DNAs are there? This is one and this is the other. So, 2 hybrid DNAs will be there. Number of DNAs with both strands N15, this is 0, because we have 2 hybrids, remaining are new type. This is the reason. Now, DNAs with both strands N14 type, new type. So, out of 8, 1, 2. 2 are hybrids, remaining 6 are new type. So, 6 new type of DNAs are there, 2 hybrids are there, there is 0 old type DNAs are there, 14 N14 strands are there, 2 N15 strands are seen here. Now let us consider one more numerical problem basing on replication of DNA. Here this is the problem. N14 DNA was allowed to replicate in N15 medium for 4 generations. Find out how many total DNAs are formed, how many hybrids are there, total strands of DNA, ratio of N14 is to N15 strands, number of DNAs with both strands N15 and number of DNAs with both strands N14. How many replications are here? 4 replications. So 2 to the power 4. So 16 DNA molecules must be there. 16 DNA molecules must be there. In these 16 DNA molecules, so total strands will be 16 into 2, 32 SS are there, single strands are there. Single strands are there, and of these the parental DNA will be present in 2 strands only. Out of these the parental DNA will be present in only 2 strands. So hybrids will be 2, where one strand is old type and other strand is new type. So 14, 15 will be 2 DNAs only, hybrids will be 2, and new types that is one strand N14, both strands N15, because this is the new medium, and out of 16, 2 are like this, remaining 14 will be like this. 14. Now, total strands. How many total DNAs are formed? 4 replications means 2 to the power 4. So 16 DNA molecules are formed. How many hybrids are there? Irrespective of number of replications, hybrids will be 2 only. This is always fixed. Irrespective of number of replications, whether the DNA undergoing single replication, 2, 3, 4, 5, whatever is the number of replications, the hybrid number will be only 2. So 2 hybrids are there, and out of 16, 2 are hybrids, and new type will be only 14. 16 minus 2, 14 will be there. How many hybrids? So this we have answered. 2 hybrids are there, and total strand DNA will be 16, and total strands of DNA, one DNA is made up of 2 strands. So 16 DNAs, 16 into 2, 32 single strands are there. Now ratio of N14 and N15 strands, in the only these 2 are hybrids, means one strand N15, other strand N14. So 2 such are there. So old ones, that is N14, 2 strands are here, whereas out of 32 strands, 2 belong to N14 and remaining 30 belong to N15 type or heavy type. So this comes to 1:15. This comes to 1:15. Number of DNAs with both strands N14 will be 14. Now let us consider one more numerical problem. N15 type of DNA was allowed to replicate in N14 medium for 3 generations. The division time is 20 minutes and generation time, that is growth time is 30 minutes. Find out how many DNAs are formed and time taken to complete the process. N15 డిఎన్ఏను N14 యానకములో 3 తరాల వరకు ప్రతికృతి చెందినట్లు చేశారు. విభజన సమయము ద్విధా విచ్ఛిత్తి సమయం 20 నిమిషాలు, అనంతరం పెరుగుదల సమయము 30 నిమిషాలైనా ఎన్ని డిఎన్ఏలు ఏర్పడినాయి, ఎంత సమయంలో ఈ ప్రాసెస్ ఈ విధానము పూర్తి అయినది కనుగొనాలి. To find this out, we have to calculate how many replications are here, how many generations. 3 generations are here. So this is the first cell, then it undergoes division, and what is the division time? 20 minutes. 2 cells are formed, then it will require growth time of 30 minutes, growth time of 30 minutes, then division time of 20 minutes division. So this will be division. At the end of that we have 4 cells. So this is division. Then again it will be subjected to growth of 30 minutes and division of 20 minutes. So each cell will be producing 2 daughter cells. So first cell 2 daughter cells, second cell also producing 2 daughter cells, third cell also producing 2 daughter cells, fourth cell also producing 2 daughter cells, and division time is 20 minutes. So at the end of 3 replications, 8 daughter cells are or 8 daughter DNA molecules are produced. So number of DNAs formed is 8. So these are, let us assume these are cells in a live organ, DNA in a live organism, and this is the case. So you have 8 DNAs are formed, and what is the time for cells? 20 plus 30, 50, 70, 100, 120 minutes. So total time for the process is 120 minutes or 2 hours. So this is 20, 30, 50, 50 plus 50, 100 and 20, 120 minutes. Now let us consider replication of DNA in semi-conservative method. Regarding replication of DNA, the DNA molecule at ORI site, ORI representing origin of replication, at ORI site the DNA opens up, at ORI site the DNA opens up and a replication fork is formed. A replication fork is formed. A replication fork is formed. So at ORI site, the replication, the DNA opens up. So the process begins at ORI site. A replication fork is formed, and certain enzymes like helicases help in unwinding of DNA. Helicases help in unwinding of DNA strand, and 𝗵𝗿isomerase help in removing tension, because at some portion the DNA is unwound and the remaining is in coiled condition. So some kind of tension is created. To remove that, topoisomerases will be acting. The two strands of DNA open up, and this is the picture. The dsDNA opens up. This is the replication fork. This is the replication fork. Two enzymes, helicases, they are responsible for unwinding of DNA, and topoisomerases, they are helpful in removing tension in the DNA. Now, let us assume that the sequence of nucleotides 𝗳𝗬𝗁𝗔 𝗳𝗔 here, and this is the first nucleotide, and the enzyme, the third and most important enzyme is DNA dependent DNA polymerase. DNA dependent DNA polymerase. This enzyme is responsible for synthesis of complementary strands. This enzyme is responsible for synthesis of complementary strands. On its own, it cannot initiate, unable to initiate polymerization. So, an RNA primer comes and lies opposite to the template strand. This is the template strand whose polarity is 3,5, and the DNA dependent DNA polymerase starts joining 𝗧𝗦𝗔𝗧 acid to the last nucleotide of RNA primer, then A, then C, then G, then Z, then C. So, this is RNA primer. To the RNA primer, the first nucleotide is joined. What is the reason for this? The DNA dependent DNA polymerase on its own cannot start the process of the synthesis of new strand. It can join the second nucleotide, but not the first one. So, an RNA primer is attached here, and to this RNA primer it will join the complementary nucleotide. T is complementary to A, and T is joined here. A is complementary to T, this is joined to T, and C is complementary to G, so C, then G is complementary to C, and like that. In this manner, this being added. So, this is called continuous strand, or this is called leading strand. This is called leading strand. There is further difficulties also there in this process. There are further difficulties also. What is the further difficulty? It can polymerize DNA dependent DNA polymerase can polymerize in 5'3" direction only. It can continue polymerization in 5'3" direction. So, what is the polarity of this new strand formed? This is 5' and this will be 3'. So, the new strand is produced in 5'3" direction on a 3'5" template. This is produced on 3'5" template. So, the template must be in 3'5" direction, and new strand synthesis is only on 𝗫3 direction. So, this is the process, and it cannot initiate synthesis from this end. So, after reaching this point, the DNA dependent DNA polymerase, let us assume, takes a turn and starts synthesizing broken fragments of DNA, and this is called lagging strand. And on 3,5 template strand, the synthesis is continuous, and here it is synthesized in continuous manner. On 𝗫3 polarity strand, the process is broken, and the broken segments are called Okazaki fragments. The broken segments are called Okazaki fragments. So, at the ORI site, origin of replication site, the DNA opens up, forming a replication fork, and for opening of the double-stranded DNA, coiled DNA, helicages are useful, and to remove tension, 𝗃𝗍𝗺𝗔𝗧 help, and DNA dependent DNA polymerase, it begins the process, but on its own it cannot initiate the process. So, RNA primer is attached. An RNA primer is attached, the new strand is synthesized attached to the RNA primer only. And so, because A is here, T will be added to the RNA primer, T is here A, G is here C, C is here G, C is here Z, G is here C. So, in this complementary manner, in complementary basis of complement or principle of complementarity, the new strand is synthesized. So, the polymerization of deoxyribonucleotides occur here. Now, the process is, so in this regard we have to bear in mind the DNA polymerase on its own cannot begin the process, that is the first thing. So, there is RNA primer. Next, it can polymerize only in 𝗫3 direction, that is the template, for that the template must be in 3,5 direction. So, the new strand is always synthesized in 𝗫3 direction, and this is 3. So, this is 5, 3, 5, 3, 5, 3, 5, 3. So, at the beginning of each we have an RNA primer. An RNA primer will be here at the beginning of each of the Okazaki fragments. DNA ప్రతికృతి అర్ధ సంరక్షక విధానంలో జరిగేటప్పుడు ORI origin of replication ప్రతికృతి ఆరంభ స్థానం దగ్గర DNA ఓపెన్ అవుతుంది. దీనికి హెలికేజ్ అనే ఎంజైమ్ తోడ్పడుతుంది. DNA ద్విపోచ DNA వాటి మధ్య హైడ్రోజన్ బంధాలు తెగటం వలన మిగిలిన భాగంలో బంధాలు ఉండటం వలన టెన్షన్ లేదా తన్యతను తొలగించడానికి 𝗃𝗍𝗺𝗫 ఎంజైములు తోడ్పడతాయి. DNA ప్రతికృతికి కావాల్సినటువంటి ముఖ్యమైన ఎంజైము DNA ఆధారిత DNA పాలిమరేజ్. ఇది DNA ప్రతికృతిని ప్రారంభించలేదు, కొత్త పోచను ఏర్పడుట సరాసరి ప్రారంభించలేదు. అందువలన మూస ఫలకానికి ఎదురుగా RNA ప్రైమర్ వస్తుంది. RNA ప్రైమర్కు కొత్త DNA పోచలోని న్యూక్లియోటైడ్లను వరుస క్రమంలో అమర్చుకుంటూ, వరుస క్రమంలో జత చేసుకుంటూ కొత్త పోచను తయారు చేస్తుంది. DNA ఆధారిత DNA పాలిమరేజ్ ఎల్లప్పుడూ పాలిమరైజేషన్ 5-3 దిశలో జరుపుతుంది. కాబట్టి మూస ఫలకం యొక్క పొలారిటీ 3-5 మాత్రమే ఉండాలి. కొత్త స్ట్రాండ్ కొత్తగా ఏర్పడే పోచ ఎల్లప్పుడూ 5-3లో ఏర్పడుతుంది. దీనిని లీడింగ్ స్ట్రాండ్ అని పిలుస్తారు. 3-5 పోలారిటీ కలిగిన ధ్రువత్వం కలిగినటువంటి DNA మూస ఫలకం మీద నిరంతరాయంగా కొత్త పోచ తయారవుతుంది. 5-3 ధ్రువత్వం కలిగినటువంటి మూస పోచ మీద ఏర్పడేటువంటి DNA ఖండాలుగా ముక్కలుగా ఏర్పడుతుంది. ఈ ముక్కలను 𝗳𝗏𝗃 ఫ్రాగ్మెంట్స్ అని పిలుస్తారు. ప్రతి ముక్క ముందు భాగంలోనూ RNA ప్రైమరు కలపబడి ఉంటుంది. Regarding replication of DNA, this is the ⅕-5 polarity, and when this is acting as template, synthesis is continuous. This is RNA primer and this is newly synthesized strand, and after reaching this point, the dependent DNA polymerase takes a turn and starts adding the DNA in broken manner, and these broken fragments are called 𝗳𝗏𝗃 fragments. So the synthesis of new strand of DNA is always in 𝜙3 direction. What is the reason for that? The template will be ⅕-5. The template shows a polarity of ⅕-5. This is the reason it is always in 𝜙3 direction. And in prokaryotic organisms, 3 kinds of DNA polymerases are there, and the one kind of DNA polymerase, that is DNA polymerase 1, is involved in DNA repair. Now, so a continuous strand is formed and a discontinuous strand is also formed. And what is brought here? Suppose I write A here, T, and what is T here, and what is this A here? A here means 𝗳𝗏𝗃 acid, or in other words it is 𝗳𝗏𝗃 monophosphate. And what is T here? This is 𝗬𝗏𝗃 acid, or thymidine monophosphate. What we generally write as T represents thymidinic acid or deoxythymidinic acid or deoxythymidine monophosphate, and A will be in DNA, A will be deoxyadenic acid or deoxyadenosine monophosphate. In cell, in cytoplasm or nucleoplasm, this will be existing as deoxy-AMP. This reacts with pyrophosphate producing deoxy-ATP. The deoxy-ATP is brought to the site here, and similarly deoxy-TMP, it exists here. This reacts with pyrophosphate producing deoxy-TTP. So the thymidine triphosphate is brought to the site here, and TMP, T means TMP remains here and pyrophosphate is released. Let me write T here, and this deoxy-ATP is brought here and pyrophosphate is released. And when G is here, deoxy-CTP is brought here, deoxy-CMP, it will react with pyrophosphate producing deoxy-CTP. This is brought here and deoxy-CMP remains here and pyrophosphate is released. The process requires lot of energy consuming process. So, the entire strand cannot be replicated in a single stretch. So, the process goes on and the replication direction is normally bidirectional replication occur. Replication is bidirectional, rarely it is unidirectional. In case of prokaryotes, number of ORI sites will be 1. In case of prokaryotic organisms, number of ORI sites will be 1. In case of linear DNA of eukaryotic organisms, many ORI sites will be there. And in case of the Drosophila fruit fly, about 3,500 ORI sites are there on 8 chromosomes of the Drosophila melanogaster or fruit fly. Normal direction of replication is bidirectional. The nucleotide triphosphates have 2 roles to perform. It has to supply the T, that is thymidyllic acid, it has to supply, and also energy for synthesis of the bond. Phosphodiester bond energy also must be supplied by the same. For that purpose, the AMP, deoxy-AMP reacts with PPI, forming deoxy-ATP, and this deoxy-ATP comes here, leaving TMP, pyrophosphate is released. The second and third phosphates present in triphosphates are energy rich. So, the bond breaking energy is used for synthesis of phosphodiester bond. So, the T joined here, A joined here, C joined here. In similar manner, deoxy-GMP reacts with pyrophosphate, producing deoxy-GTP. And again, you have addition of deoxy-guanilic acid here, and it not only supplies the triphosphates or the nucleotides, not only supply the nucleotide, but also supply energy for bond formation. Once this process is completed, the RNA primers are removed. And the gaps between 𝗂𝗃 fragments are filled by DNA ligase. DNA ligase fills the gaps between 𝗂𝗃 fragments by adding appropriate nucleotides depending on the principle of complementarity. Basing on principle of complementarity. DNA ప్రతికృతి చెందినప్పుడు RNA ప్రైమర్కు డిఆక్సిరైబోన్యూక్లియోటైడ్లు జత చేయబడతాయి. మూస ఫలకములో ఏ ఉన్న టీ అనేది కొత్తగా ఏర్పడే పోచలో చేరుతుంది. ఏ అంటే ఏమిటి? ఏ అంటే డిఆక్సీ ఎడినిక్ ఆమ్లము లేదా డిఆక్సీ ఎడినోసైన్ మోనోఫాస్ఫేట్. టీ అని ఉపయోగించేది, టీగా పిలువబడేది థైమిడిలిక్ ఆమ్లము లేదా థైమిడైన్ మోనోఫాస్ఫేట్. అదేవిధంగా జీ అని పిలువబడేది డిఆక్సీ గ్వానిలిక్ యాసిడ్, సిగా పిలువబడేది డిఆక్సీ సైటిడిలిక్ యాసిడ్. ఇవి ట్రైఫాస్ఫేట్ల రూపంలో కేంద్రక రసంలో లేదా కణద్రవ్యంలో ఉంటాయి. ఇవి 2 విధులను నిర్వర్తిస్తాయి. నత్రజని క్షారము లేదా న్యూక్లియోటైడ్ను సరఫరా చేయటం, న్యూక్లియోటైడ్ల మధ్య ఫాస్ఫోడైస్టర్ బంధాలు ఏర్పరచడానికి కావలసిన శక్తిని సమకూర్చటం. ఈ పని చేయడానికి డిఆక్సీ-AMP పైరోఫాస్ఫేట్తో చర్య పొంది డిఆక్సీ-ATP, అదేవిధంగా డిఆక్సీ-TMP పైరోఫాస్ఫేట్తో చర్య పొంది డిఆక్సీ-TTP, CMP చర్య పొంది డిఆక్సీ-CTP, అదేవిధంగా డిఆక్సీ-GMP చర్య పొంది డిఆక్సీ-GTP ఏర్పరుస్తాయి. ఈ ఒక్కొక్కటి ఈ స్థానంలో వచ్చి మోనోఫాస్ఫేట్ను వదిలి పైరోఫాస్ఫేట్ అంటే ఒకటి డిఆక్సీ-TMP మరియు పైరోఫాస్ఫేట్ రెండవ ఫాస్ఫేట్ బంధము విచ్ఛిన్నమవటం వలన విడుదలైన శక్తిని ఉపయోగించుకొని ఫాస్ఫోడైస్టర్ బంధాలను ఏర్పరుస్తుంది. ఈ విధంగా కొత్త పోచ ఏర్పడటానికి న్యూక్లియోటైడ్ ట్రైఫాస్ఫేట్లు రెండు రకాలుగా పనిచేస్తాయి. ఒకటి న్యూక్లియోటైడ్ను సమకూర్చుట, రెండు బంధాలు ఏర్పరచడానికి అవసరమైన శక్తిని సమకూర్చుట. ఈ విధంగా కొత్త పోచలు ఏర్పడతాయి. కొత్త పోచలు ఏర్పడిన తర్వాత RNA ప్రైమర్లు తొలగించబడతాయి. అదేవిధంగా ఖండాలుగా ఏర్పడిన ఒకజాకి ఫ్రాగ్మెంట్స్ వీటి మధ్య DNA న్యూక్లియోటైడ్లు డియాక్సీరైబోన్యూక్లియోటైడ్లను DNA లైగేజ్ చేర్చటం వలన నిరంతరాయంగా ఉండే కొత్త పోచ నిర్మాణం కనబడుతుంది. ఇక ప్రతికృతి ఏ దిశలో జరుగుతుంది? అరుదుగా ఏకదిశలో ప్రతికృతి జరుగుతుంది. సర్వసాధారణంగా ప్రతికృతి ద్విదిశలో బైడైరెక్షనల్గా ఉంటుంది. వలయాకార DNA కలిగిన కేంద్రక పూర్వజీవుల్లో ప్రతికృతి చెందటానికి ORI సైట్ ఒకటే ఉంటుంది. నిజ కేంద్రక జీవుల యొక్క రేఖా రూపంలో ఉండే క్రోమోజోముల్లో అనేక ORI సైట్లు ఉంటాయి. మరలా దిశ ప్రతికృతి కనబడుతుంది. Now let us try to conclude what we have discussed so far. RNA world. RNA is the first genetic material. By undergoing chemical evolution, it has produced DNA which is more stable. Some important processes like protein synthesis, splicing are centered around RNA because it is the first genetic material. Regarding replication of DNA, the double helical structure of DNA was proposed by Watson and Crick, and due to presence of fixed complementarity between adenine and thymine and also guanine and cytosine, Watson and Crick proposed that the DNA replication occurs in semi-conservative manner. The DNA replication occurs in semi-conservative manner without any experimental proof. The experimental proof towards this, regarding this, was supplied by Messelson and Stahl using N15 NH4Cl or heavy nitrogen to observe whether replication of DNA occurs in semi-conservative manner or other way round, and this was proved by them in 𝗧𝗦𝗃 coli, a prokaryotic organism. In case of eukaryotic organisms, Taylor and his associates, using radioactive thymidine in 𝗬𝗂 faba roots where mitotic division occur, they have proved that the replication of DNA in eukaryotic organisms is also in semi-conservative manner. The replication of DNA requires certain enzymes, and the enzymes are helicases, and all these helicases are also called gyrases, topoisomerases, and also DNA dependent DNA polymerase. DNA dependent DNA polymerases new strand or nucleotides in 5'3" direction, and because of this the template must be in 3'5" direction. The DNA polymerase cannot initiate the process on its own, so it adds new nucleotides to an RNA primer, and the new strand produced on 3'5" polarity strand, template strand, is continuous, that is called leading strand. On the other strand of DNA that shows 5'3" polarity, it is synthesized in broken manner called 𝗂𝗽𝗃 fragments. At the end of the process, the 𝗂𝗽𝗃 fragments are joined by DNA ligase. The nucleotide triphosphates of ATGC, they play two roles. One is supplying the nucleotide and also bringing the energy required for bond formation. With this, let us conclude. Thank you.